Hello friends, welcome to my channel Helping Hand for Nurses. This video is the second part of the subject microbiology. So in this video I have included morphology of the bacteria, classification of bacteria according to morphology and on various aspects and the structure of the bacteria. And this video is very important, and most of the questions which has asked in nursing exams is from this part, that is a structure of the bacteria and the morphological classification of the bacteria. So please watch the full video, and if you like this video, please like and subscribe it. Let us start the video. Classification of bacteria. Bacteria can be classified on various aspects. So first one is the morphology. So bacteria is classified on the basis of morphology or on the basis of shape. So on the basis of morphology, bacteria is classified into coccus, bacillus and spirilla. So coccus or cocci, it is the plural form, bacilli is the plural form, and singular form is the bacillus, and this is spirilla. So coccus are round, oval or spherical shaped bacteria, and bacillus are rod shaped bacteria, and spirals are helical or spiral shaped bacteria. So coming to the coccus, monococcus are bacteria that exist as a single spherical cell. If the cocci, it exists as a single spherical cell, then it is called monococci. And if the cells are arranged in pairs after cell division, then it is called diplococci. If the cells are arranged in pairs, then it is diplococci. So single spherical cell, it is the monococci, and paired cell, it is a diplococci. Example is Streptococcus pneumoniae. And if the cocci are joined in a plane and arranged in a chain pattern, then it is called streptococci. If the cells are arranged in a chain-like pattern, then it is called streptococci. Example is Streptococcus pyogenes. Coming to tetrad, here the tetrads are arranged in a group of 4 cells. Tetrads are arranged in a group of 4 cells. An example for tetrads are Micrococcus species and Pediococcus. Staphylococcus, here the cells are arranged in an irregular cluster. That is, it has a grape-like structure. Example is Staphylococcus aureus. Next is Sarcina. So Sarcina bacteria are anaerobic gram-positive bacteria. Anaerobic gram-positive bacteria, and it occurs as a group of 8 cells. Example is Sarcina ventriculi. So coming to the coccus, coccus is again divided into monococci, diplococci, streptococci, tetrad, staphylococci and sarcina. Monococci, here single spherical cell is there, and diplococci, pair of cells is there, pair of spherical cell is there, and streptococci, here the cocci are arranged in a chain-like pattern, and here in tetrad we can see 4 cells, and in sarcina it is combined of 8 cells, and staphylococci we can see grape-like structure. Then coming to the bacillus, bacillus are single unattached cell that it looks like a rod. So bacillus are single unattached cell and it looks like a rod. An example for bacillus is Bacillus cereus and Salmonella enterica. Bacillus cereus and Salmonella enterica are example for bacillus. Coming to diplobacilli, here the 2 rods are attached to each other and found in pairs after cell division. Here the 2 rods are attached to each other and they are found in pairs. It is called a diplobacilli. An example for diplobacilli is Moraxella bovis and Klebsiella rhinoscleromatis. Klebsiella rhinoscleromatis are examples for diplobacilli. Coming to streptobacilli, same as in that of streptococci, here the bacilli are arranged in the chain-like pattern. That is, due to cell division in one plane, bacilli are arranged in a chain. Example is Streptobacillus moniliformis and Streptobacillus felis. Next is Coccobacilli. These are short compared to other bacilli and it is oval in shape. They appear just like a coccus. So it is known as Coccobacilli. Examples are Chlamydia trachomatis, Haemophilus influenzae. So Chlamydia trachomatis and Haemophilus influenzae are Coccobacilli. Next is palisades. Here the bacilli after cell division bend and therefore arranged in a palisade or a fence-like structure. Here the bacilli are arranged like a fence-like structure. Example is Coryn bacterium diphtheria. So these are the arrangement of the bacilli, that is bacillus, diplobacilli, here pair of cells is there, streptobacilli, chain of bacilli is there, and cocobacilli, that is oval shaped structure, oval shaped bacilli, and palisades, here the bacilli is in the form of a fence. Coming to the spirilla, here these bacteria are spiral or helical in shape. Based on the thickness, flexibility and motility of the cell, these spirilla are again divided into spirochetes and spirillum. Example for spirochetes are Leptospira and Treponema pallidum. So example for spirochetes are Leptospira and Treponema pallidum. Spirochetes are spiral and thin and flexible. And coming to spirillum, they are gram-negative bacteria and they have external flagella and they are helical shaped or corkscrew form. Example is Campylobacter jejuni and Helicobacter pylori. Campylobacter jejuni and Helicobacter pylori. Next is a vibrio, which is a comma shaped structure. It appears like a comma. And these are mostly gram-negative bacteria. They are known to cause various foodborne diseases. Examples are Vibrio cholerae and Vibrio parahemolyticus. These are the budding and appendaged bacteria. Budding and appendaged bacteria. They can be non-motile or motile with the flagella. And another one is the filamentous. It has a long filamentous structure. Example is Candidatus savagella. Thumbprint appearance can be seen in case of Bordetella pertussis. That is, when we see this Bordetella pertussis under the microscope, we can see a thumbprint appearance. This is very, very important. You should keep in mind that the thumbprint appearance is seen in Bordetella pertussis. For which bacteria? It is seen in Bordetella pertussis. Coming to the Mycobacterial leprae, we can see cigar bundle appearance, that is bundles of cigar appearance. And coming to the Clostridium tetani, we can see drumstick appearance. So drumstick appearance is seen in Clostridium tetani. Bamboo stick appearance is seen in Bacillus anthracis. Bamboo stick appearance is seen in Bacillus anthracis. Chinese letter arrangement can be seen in Coryn bacterium diphtheriae. Chinese letter arrangement can be seen in Coryn bacterium diphtheriae. Vibrio cholerae, we can see comma shaped bacteria. So if we are looking these organisms under the microscope, we can see the comma shaped structure in Vibrio cholerae. And in case of Coryn bacterium diphtheria, if the bacteria is Coryn bacterium diphtheria, we can see Chinese letter arrangement. And in case of Bacillus anthracis, we can see bamboo stick appearance. And in case of Clostridium tetani, we can see drumstick appearance. And Mycobacterial leprae, we can see the bundle of cigar appearance. And in case of Bordetella pertussis, we can see the thumbprint appearance. And in case of Yersinia pestis, we can see safety pin appearance. Safety pin appearance. So these are the classification of bacteria according to their shape. Then according to staining they retain, the bacteria can be classified into gram-positive and gram-negative. So after staining, gram-positive bacteria will appear blue, purple or violet in color. And in case of gram-negative, the bacteria will appear red or pink in color. On the basis of nutrition, bacteria is classified into autotrophic bacteria and heterotrophic bacteria. Autotrophic bacteria means they can prepare their own food, and heterotrophic means they depend on others for food. On the basis of flagella, this bacteria is divided into Atrichus, Monotrichus, Amphitrichus, Lophotrichus, Peritrichus, Cephalotrichus. This is very important. On the basis of flagella, bacteria is divided into Atrichus, Monotrichus, Amphitrichus, Lophotrichus, Peritrichus and Cephalotrichus. So in case of Atrichus, Atrichus. A means absent, that is there is no flagella. Example for atrichus is lactobacillus. Lactobacillus. Coming to monotrichus, there is a single pole flagella is there. Single pole flagella is there. One flagella is at one end of the bacteria. That is a monotrichus. Example is Vibrio cholerae. So Vibrio cholerae bacteria are monotrichus. Amphitrichus bacteria contain a single flagellum on each of their two opposite ends. That is, it contain a single flagellum on each of their two opposite ends. And example for Amphitrichus is Alcaligens fecalis. Next is Lophotrichus. Here we can see a tuft of flagella on one end of the bacteria. At one end of the bacteria. Tuft of flagella at one end of the bacteria or at one pole of the bacteria. Example for Lophotrichus is Spirillum. Peritrichus, here we can see the flagella all around the bacteria. That is Peritrichus. Example for Peritrichus is Salmonella typhi. Cephalotrichus, here we can see several flagella on both the end of the bacteria. Example for Cephalotrichus is Pseudomonas. So atrichus, monotrichus, atrichus absent of flagella, monotrichus single flagella at one end, amphitrichus flagella, one flagella at each end, and cephalotrichus several flagella on both the end of the bacteria, and peritrichus flagella all over the body, means flagella all over the body of the bacteria. And Lophotrichus, here the tuft of flagella will be present only at one end of the bacteria. So example for atrichus is Lactobacillus, monotrichus Vibrio cholerae, amphitrichus Alcaligens fecalis, Lophotrichus Spirillum, and peritrichus Salmonella typhi, cephalotrichus Pseudomonas. Then according to heat tolerance, bacteria is divided into psychrophilic, mesophilic bacteria and thermophilic. Psychrophilic, it can survive on extreme cold temperature. Mesophilic bacteria, it can survive on moderate temperature. Thermophilic bacteria, it can survive on hot temperature. And according to pathogenicity, it is further divided into pathogenic bacteria and non-pathogenic bacteria. Pathogenic bacteria which causes diseases and non-pathogenic which does not cause any disease. Structure of bacteria. So you know that bacteria is unicellular and it is made up of following structures like capsule, cell wall, plasma membrane or cytoplasm membrane, cytoplasm, ribosomes, plasmid, nucleoid, then flagellum and pili. So first one is a capsule. So a capsule is gelatinous secretion of bacteria which will provide a protective covering for the bacteria. So capsule is gelatinous secretion of bacteria that makes a protective covering for the bacteria, and every bacteria does not have this capsule. Some of the bacteria are non-capsulated. And these capsulated bacteria are non-motile, that is it cannot move. Capsulated bacteria cannot move. Next one is the cell wall. So cell wall is a tough and rigid structure surrounding the bacterium like a shell. It is a tough and rigid structure surrounding the bacterium like a shell. And the function of the cell wall is that it provides shape to bacteria, rigidity to bacteria, support to bacteria, and it helps in division in bacteria. And one important point is that in gram-positive bacteria, this cell wall is more thick. In gram-positive bacteria, cell wall is more thick. And another point is that in gram-positive bacteria, cell wall is composed of peptidoglycan. Gram-positive bacteria, cell wall is composed of peptidoglycan. Whereas in gram-negative bacteria, cell wall is composed of lipopolysaccharide. So in gram-positive bacteria, cell wall is composed of peptidoglycan, and in gram-negative bacteria, cell wall is composed of lipopolysaccharide. In case of acid-fast bacilli, we can see mycolic acid over cell wall. In acid-fast bacilli, we can see mycolic acid over cell wall. And coming to the smallest non-bacteria, which is the smallest non-bacteria? It is Mycoplasma. Mycoplasma is the smallest bacteria. It has no cell wall. So the bacteria without a cell wall is a Mycoplasma. It is the smallest non-bacteria. So that is regarding the cell wall. So the important points in the cell wall is that in gram-positive bacteria, cell wall is composed of a peptidoglycan, and in gram-negative bacteria, it is composed of lipopolysaccharide. And in acid-fast bacilli, we can see mycolic acid in cell wall, and Mycoplasma is the smallest bacteria and it does not have any cell wall. Next is the cell membrane or the plasma membrane. So it is a thin elastic membrane which separates the cell wall from the cytoplasm. And the functions of the cytoplasm is that it synthesis of cell wall and provide mechanical strength to the bacteria. And also it helps in DNA replication. Then coming to the cytoplasm, this is cytoplasm. So it is a suspension of organic and inorganic solids in a watery solution. And it has a structure like ribosomes and plasmid. And ribosomes are granules distributed in whole cytoplasm and they are responsible for protein synthesis. In prokaryotes, we can see 70S ribosomes and in eukaryotes, we can see 80S ribosomes. Then coming to the nucleode, we know that bacteria has no nucleus and there is no nuclear membrane or nucleolus. And the nucleode, it has a DNA with up to 3500 genes. DNA with up to 3500 genes and DNA is circular. And plasmids are small circular DNA with 5 to 100 genes. Plasmids are small circular DNA with 5 to 100 genes. Coming to the flagellum or flagella, they are cytoplasmic appendages protruding through cell wall. Cytoplasmic appendages protruding through cell wall. These are the thread-like structure composed of protein flagellin. This flagellum is composed of a protein called flagellin. And it is the organ of motility. So organ of motility in bacteria, which is the organ of motility in bacteria? It is the flagellum or flagella. Then another structure is that pile. Pile, it is mostly seen in gram-negative bacteria and it is a hair-like tubular microfibers. And the function of this is to attach the cells to the surfaces. And to transfer DNA to another cell. And another structure is fibrae, which is not shown in this diagram. Fibrae, these are bristle-like short fibers. It is present in both gram-negative and gram-positive bacteria. But pile is present only in gram-negative bacteria, but fibrae is present in both gram-negative and gram-positive bacteria. These are bristle-like short fibers. And the main function is the attachment to the host surface. So these are the important structures of the bacteria.